Hello, everybody. Um, to, with this video, we're going to finish glycolysis, and we're going to hopefully try to explain why the glycolytic reactions we saw in the previous videos are the way they are. In other words, we're going to be talking about the evolution of glycolysis. This is pretty cool, and we're going to try to do this for a few pathways. I think the case is very convincing for glycolysis. It's actually very gratifying to see all these reactions. They seem disconnected, and why they are the way they are. And I think we can answer this question. This is not in textbooks yet. I think eventually it will make it, and hopefully you will enjoy it. All right. So who came first in terms of organisms? Are the ones that made their own food, or the autotrophs, or the ones that rely on pre-existing organisms so they can eat them and get the food they need? These are important questions to answer. And in that context, uh, who came first, glycolysis or gluconeogenesis? Now, of course, autotrophs came first. Okay, there were the ones before photosynthesis. They were living off the chemistry basically on the planet, which we still see today. By the way, deep in the oceans, next to thermal vents that come out of the crust of the earth. I mean, you find an incredible variety of microbes that do weird chemistry, and they are able to make their own food without relying actually on much on anything else. So it makes sense that the autotrophs came first. We know that from the geological record, um, and current evolutionary analysis of genomes and so on and so forth. So in that sense, then, if autotrophs came first, it's gluconeogenesis that must have come first, not glycolysis. I mean, there was no glucose around for the autotrophs, right? And that will become important. Now, the pathway is universal. Every organism on the planet does glycolysis. The question you can ask and answer these days by examining the sequences of the genes that encode these enzymes is how many origins are there for each enzyme? How many times have they been discovered, evolved in the course of evolution over the billions and billions of years? Is it one? Is it more than one? Uh, and that's the answer we can get. So it turns out that, let me move myself a little bit, 4 out of the 5 enzymes in phases 2 and 3, so the lower part of glycolysis, have a single origin. Have a single origin. So some organism, some ancestor of all of us hit on it at some point. It was very good, and basically after that it was fixed, and you can't improve upon it, right? So and everybody followed the same strategy after that. In contrast, if you look at the upper part of glycolysis, where we break down glucose to two trioses, that has multiple origins. So uh, even in the least evolved organisms, so there are many ways to skin the cat, so to speak, for the upper part of glycolysis, which suggests that trioses are really the main intermediates. And in fact, that's what we see. When we look at biosynthesis, you have 3-phosphoglycerate. I told you it's a biosynthetic block that's 3 carbon units. You can make serine out of it and other amino acids. Phosphoenol pyruvate, also a 3 carbon block. You can make aromatic amino acids and others. You have glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. These are key intermediates for lipids. So, but we don't see that for the 6 carbon units of the upper part of glycolysis. So making them seems the most ancient process. So all this again suggests that autotrophy and gluconeogenesis came first. Okay, the first autotrophic organisms, they had um, to to make these important trioses. So probably they were running some of these reactions in reverse. Okay, so that's gluconeogenesis. Furthermore, the upper part, there is no obvious reason why it should culminate with two trioses, so 2 3 carbon units, unless of course the 3 carbon units was a pre-existing module, but then the upper part had to fit in somehow. Because chemically speaking, you can, instead of splitting a 6 carbon unit to 3 plus 3, you can have a 4 plus 2. splitting. It's quite simple. Okay, you get a 5 carbon unit and you get a 4 carbon unit. Sorry, and a 2 carbon unit, which immediately gives rise to glycolaldehyde. So that's perfectly reasonable, perfectly fine, chemically speaking. There is no reason why it wouldn't be that way, but it isn't. And so the reason that it isn't that way chemically is because the pathway has to fit to the triose, more ancient module that you have in lower glycolysis, as we described. So, all right, so the lower part then becomes very important. And the upper part, as we see, actually comes in many flavors, not just one. It's not fixed, and we'll discuss at the very end a couple different, actually one more way that the upper part can take place. But the lower part is fixed. So the question is, these 5 reactions, why is it? Why are they the way they are? Okay. So for that, let me switch to. We have to do some notes on the board. So let me switch to the whiteboard, and we'll get back to the slides later. So we'll share screen, share a different screen. We'll share this. Share. Let's make another one. New whiteboard. Okay. So here we go. All right. So how does it start? The lower part of glycolysis. It starts with, let's say, with glyceraldehyde 3-phosphate. So let's write it out. We'll put it right in the middle here. I'm going to skip all the protons for now. We'll get to some of the protons later. So the point here is to go from here to let's say to lactate or some other 3-carbon equivalent 3-carbon unit, and so let's see. So lactate will be so So we consider here um, um, glycolysis and fermentation. So the reason is important because again, it's only with fermentation that glycolysis can keep running, okay? Because you can regenerate the NAD+. So this is glyceraldehyde 3-phosphate at the top. This is lactate. How can we go there by simple redox reactions, okay? So let's see, all internal within the molecule, right? So we can imagine that the last carbon stay the way it was. So carbonyl, carbon, carbonyl. The next carbon becomes oxidized to become a carbonyl, and the one next to it, from a hydroxycarbon, it just becomes a hydrocarbon. So that's an internal redox. So we couple a reduction of this carbon with an oxidation of the one next to it. Then we can go here. In the same way, we couple now the oxidation of the original carbonyl carbon on glyceraldehyde 3-phosphate to a carboxyl carbon, and the newly created carbonyl carbon gets reduced to hydroxycarbon. Or we can go directly from glyceraldehyde 3-phosphate to lactate, right? What is the let's calculate the delta E. So if we can figure out the chemistry, so this is like a exercise on paper. If we can see whether this is thermodynamically feasible, and then we'll figure out if there is any chemistry that will allow us to do these reactions and see if that's possible. Because if it is possible, we can get, why have 5 reactions when we can get to glyceraldehyde 3-phosphate to lactate in 1 reaction or maybe 2? Okay, why do we have the 5? Of course, the 5 gives us 2 ATPs. Do we get the same thing from this chemistry? In principle, all right? So how do we calculate it? So let's name this reaction 1. So we can get confused, 2 and 3, okay? And let's look at the reaction 1. The delta E will be, oh, and again, just to remind you, let's write, let's just remind you again the oxidation states of carbon. We have a hydroxy carbon plus 2 electrons will give us hydrocarbon, and that's, let's say, plus 100 in millivolts, the E value, okay? If it's a carbonyl carbon, that will be the half cell to give us the hydroxy carbon. That's minus 250. And if it's a carboxyl carbon, to give rise to a carbonyl carbon, that's, let's say, minus 250. That's minus 500, more or less. All right, so let's go to reaction 1 now. What carbon got reduced? Because from that E value, we'll subtract then whatever got oxidized. So it is the terminal carbon here, okay? It was a hydroxy carbon, and it got reduced to hydrocarbon. So the E of that is 100. So let me just point again. So this got reduced, okay? And the other one got oxidized. So if we get to the E values, we start with whatever got reduced, and that's plus 100. Actually, and then whatever got oxidized, that would be this reaction. That's minus 250. So that would be, plus 350, which is thermodynamically favorable. And it's, for 1 ATP, I forgot to mention here, 1 ATP, you need about a plus 250 delta E to drive ATP synthesis. So that reaction, actually, 1 will give you 1 ATP. Likewise, if we go to the second reaction, now what got, let's see, what got reduced? We were here. So it's this green carbon now got reduced. Okay, so it's this one, the second reaction that we're dealing with. A carbonyl carbon, we start with a carbonyl carbon and it gets reduced to hydroxycarbon. So that's minus 250 minus what got oxidized, which is this carbon right here. It became a carboxyl carbon. That's minus 500. So that's 500 minus 250. That's a plus 250. So that gives us also just about 1 ATP. So the conclusion is, if you start from glyceraldehyde 3-phosphates on here at the top, right there, and you go to 2 reactions, this one and this one, you do get 2 ATPs out, okay? 1 and 2. If you go straight from glyceraldehyde 3-phosphate all the way to lactate, you get delta E. Again, what got reduced? The last carbon here, the hydroxycarbon, got reduced to hydrocarbons. That's plus 100 minus this one. Okay, so what got oxidized was the carbonyl carbon to a carboxyl carbon. That's minus 500. So that's 600 plus 600. So that can actually give you 2 ATPs. All right. So the conclusion from all this, okay, is that um, uh, uh, with one or two reactions, okay, with some internal redox reactions, one or two of them, you can recover the 2 ATPs that we recovered with 5 reactions that we saw in the lower part of glycolysis. So why bother with a 5 when one or two can do it? In fact, we can get even a little fancier here. So let's start another seat, and you will see what I mean. So we start again with um, glyceraldehyde 3-phosphate, right? And let's say we instead of starting with a with this carbon at the terminal, and why don't we pick up the one in the middle and do the following? Say this one now gets reduced, the carbon 2 gets reduced, so it becomes a hydrocarbon, and the other one gets oxidized to become a carbonyl. Okay. Remember what we did before in reaction 1 was something like this. So the question is, why number 1 or number 4? They're both equally likely, chemically speaking. Okay. And much in the same way, I mean, this one now can go on in, let's say, reaction 5, hypothetical reaction 5, and become um, this one. This carbon gets oxidized, and the other one, the carbon becomes reduced. So instead of lactate that we got here we have a 3-hydroxypropionate. Okay, so as opposed to lactate. So why do we have lactate and not 3-hydroxypropionate? I mean, it's the same principle. And by the way, we can also drive number 6 reaction to go in one reaction straight from glyceraldehyde 3-phosphate to 3-hydroxypropionate. So the reactions you see, so this was reaction 1, 2, and 3 in the previous whiteboard that we had. And here's 4, 5, and 6. The only difference is which carbon participate in the internal redox reactions. Okay, so both are, chemically speaking, just as good. If you do the calculations 4, 5, and 6 with the delta A's that we did in this scheme, okay, you will see that all of them are also positive. And from regardless of what you do, whether you do 1, 2, 3, or 4, 5, 6 that we described in the previous whiteboard, that's enough to get you the 2 ATPs. So this will give you 1 ATP, and you can calculate the delta E's as we did for the reactions on the left, 1, 2, 3. You can calculate them here on your own. That will give you another 1 ATP, and that will give you 2 ATPs. Okay, so both feasible, theoretically speaking, not feasible, but they're theoretically speaking, they're fine. The chemistry works with only 1 or 2 reactions on paper. Okay, we don't know how it works in real life, and you get the expected energy output from the lower part of glycolysis. So any one of these schemes would work. 1, 2, 3, 4, 5, 6. We have 6 ways that it could happen with fewer reactions, 1 or 2 reactions. Okay, so why the 5? So, all right, let's go through them and see what happens. For that, I will need let me just okay. I will need another whiteboard. All right. So let's start with glyceraldehyde 3-phosphate. Is it chemically possible to get this? So here we're gonna draw actually the protons. Okay. This is glyceraldehyde 3-phosphate. And it turns out that all the chemistry in all these hypothetical reactions that we discussed begins with dehydration of glyceraldehyde 3-phosphate. How does that happen? Well, so let's say you have this and this, okay? Of course, the oxygen will pick up this proton, okay? So what's going to happen? You have a dehydration, and then let's see what we got. Okay, you have this intermediate that really does, it's very unstable, but does ketoenol tautomerization. So it goes on the next one. And you get the methyl glyoxal here. Okay, so this is exactly the same as reaction one that we discussed. So the output, let's go back and see this just for 1 second. Let's pick this scheme. So what I just showed you is the mechanism to go to reaction one. Okay, we dehydrate 3-phosphoglycerate, and we get this product, okay, which is methyl glyoxal. All right, so that's possible to get actually. So already we are hitting our chemistry just fine. Everything seems to be working. So that would be reaction one. Okay, let's do another dehydration and see where that takes us. All of them again begin with dehydrations. We just pick and choose a different proton to leave, and now it's going to be this one. And okay, here we have now something very similar. Again, ketoenol tautomerization, and we end up with, let's see what we end up with. Okay, so this is called malonyl dialdehyde. And I believe that was reaction 4. Okay, let's go back to our scheme just to make sure. Of the theoretical better outcomes, we saw how reaction 1 gives this to methyl glyoxal, and now this is what we got. Okay, with another dehydration, we get the output of reaction 4. So, and all of them give the expected ATPs in much shorter sequence of reactions. Let's continue here, okay, in the dehydrations. Is that all we can do? It turns out we can do one more. Start again. With glyceraldehyde 3-phosphate. Okay, so let's pick this proton. We haven't picked this proton, so we'll take one at a time here. Let me just, these are protons here. All right, so this happens. This moves this way. Okay, we lose this proton, and then this hydroxide group will leave, of course. And goes. So you lose the water. Now we get a weird compound. It's called a ketene. Okay, and then if you hydrate that back, you actually get the straight up product from um, um, which is, yeah, we can put all the protons, which is 3-hydroxypropionate, which was, of course, reaction number 6. Okay, so let's go back now just to make sure. So it turns out this is possible, this is possible, and this is possible too. So everything seems to be happening here. And again, in terms of yield of ATP, the chemistry is very simple, very straightforward. Okay, dehydration of glyceraldehyde 3-phosphate can get you to where you want. So why not? All right. Well, it turns out. Oh, I should say. Sorry. Let's go back. So for reaction 3, there is no known mechanism. So we have no evidence. There is no example and no conceivable mechanisms how you can get from glyceraldehyde 3-phosphate straight to lactate. Okay, you can go in 2 steps. You can go to methyl glyoxal that we just described with dehydration, and then to lactate. But actually, the reaction 3 doesn't seem to happen. But all the other steps there are examples, okay, in chemical examples. So why don't they happen? Already pre-existing reactions. Now we'll switch to, so I hope that makes sense. Okay, hold on, let's review again. Let's summarize. We saw the possibilities of the existing chemistry, okay, how we can get these intermediates, okay, that we discussed before. We can get methyl glyoxal, we can get malondialdehyde, and we can get 3-hydroxypropionate. We saw how regardless you go through lactate shown here or 3-hydroxypropionate, how you can calculate all the delta E's, and all of them are delta E positives and enough to give you 1 ATP here or 2 ATPs for reaction number 3. And the same things happens for 4, 5, and 6, okay, the alternative plan. So why not? All right, so let's switch again to the slides. Share this. All right, and all the reactions I drew, they're all in a paper that I have linked. I mean, it's available, and you can find and read on yourself. I just on your own. I just try to explain it in simple ways. This is what I was drawing earlier. You have option A, option B. Whether you go through lactate at option A, so that would have been reaction 1, 2, and 3 here. Or you can go through the formation of 3-hydroxypropionate. Again, chemically speaking, they seem the same. It's just which carbon, you do the first redox, the internal redox reaction. And thermodynamically, again, you get enough just from the coupling of these reactions to get you the ATPs you would expect from the lower part of glycolysis, 2 ATPs, regardless of which one you do. Okay. We went through the chemistry, and as I told you, reaction number 3 here from glyceraldehyde straight to lactate, and here you see the redox states of the carbon. Okay, but reaction 3 is not possible directly, but all the others appear to be possible, and we did them on the board. So why don't they happen? And these are the the products, methylglyoxal, malondialdehyde, and 3-hydroxypropionate, which are perfectly fine, chemically speaking, but biologically speaking, every single one of them is a poison. Okay, so why is it a poison? They cross-link. So you have always, whenever you have two carbonyl groups next to each other or one carbon apart, they are really nasty. I think I have a slide. Yeah. So it will pick up, let's say, amino groups from this is methyl glyoxal, but it's the same thing for the others. Amino groups, whether they're on nucleotides, whether they're on your proteins, on lysins, for example, or whatever, and they will cross-link them. So things that are not supposed together, they're going to be essentially frozen and cross-linked, and you're dead. So even though inorganically thinking about it on pure chemical terms, all these rearrangements and dehydrations of glyceraldehyde 3-phosphate can happen, that doesn't mean they should happen in a cell. They are incompatible with life. All right. So what would the cell do then? I mean, is it stuck now? There is nothing it can do? Well, you have to add extra reactions to avoid toxic intermediates. So it does not want to go through these intermediates because they're poisonous. So the approach that most cells use is to use NAD. I mean, we didn't see any NAD, in the previous reactions, okay? But here we see it. We use NAD to move the electrons on and off, and instead of doing them internally, as we just discussed, and that eliminates, bypasses the toxic intermediates. So things can happen, but of course you have to add a couple more reactions, okay? So the reason for using NAD are two steps, okay, in glycolysis and fermentation. One is glyceraldehyde 3-phosphate dehydrogenase in GAPDH, and then we see again in fermentation when we go from pyruvate to lactate. Okay, that's where the electrons leave NADH, and you can regenerate NAD plus, and the whole thing can recirculate here. So the reason for doing this, again, for invoking these reactions, the NAD-dependent reactions, is to avoid the toxic intermediates, and that's the minimum reactions you can have, okay, to complete the lower part of glycolysis and fermentation. So it's what we see is the best possible outcome. Okay, nobody has been able to come up with a better outcome. And if you think about the rationale in these terms, the lower part of glycolysis makes perfect sense. Now, the alternative to go to 3-hydroxypropionate is actually very similar, and it depends on how you make the enol pyruvate. If you just switch a little bit where the double bond is, you can get to 3-hydroxypropionate. It turns out, so you get this enol oxopropionate as opposed to enol pyruvate. So this is it requires extra stabilization. I mean, we know that enol pyruvate is an unstable high energy compound. This is even more so, and it happens, but it's very tricky to do in aerobic condition. But it does happen in nature actually, and you do get in some unusual organisms, you do get 3-hydroxypropionate. So this chemistry is perfectly fine. Again, you invoke NAD, okay, and I should say that the ATP that you get in fermentation, the delta E value that you get in fermentation is not enough to give you ATP. That's why we don't get ATP in fermentation. It's about 100 millivolts, so it's positive. It's thermodynamically favorable, but it's not enough of a gap to get you one ATP. Instead, we get the two ATPs with the previous reactions, the phosphoglycerate kinase and GAPDH. But of course, again, we need fermentation so we can regenerate NAD+. But anyway, all these things are option A is by far the most common, okay, which is what we do. Option B, leading to 3-hydroxypropionate, does happen in nature in some cases. Okay. So the most common one is the best possible way. As smart as you may think you are, and people have thought about this, they cannot come up with a better solution. All right, so this is the overall scheme. Then again, all the reactions in glycolysis, and we discussed how the lower part makes sense. Okay, um, and if we couple this with the upper part of glycolysis, why do we have every single reaction the way it is? Okay, the first one, we have to phosphorylate to keep glucose inside. That's hexokinase. Then we have to convert the aldose to a ketose. That's a fructose to make fructose. And that's important because you won't be able to cleave the 6 carbon unit to 2 carbon units to 3 carbon units later with aldolase. So that activates aldolase. That's important. But before you can do the cleavage, okay, you have to stick another phosphate. That's PFK. So both trioses are marked at the very end with a strong negative charge, and they can never get out of the cell. So you keep them to yourself. Very important. Triose phosphate isomerase is really, it's an easy interconversion of the 3 carbon units. So if you want, you can get all of it into glyceraldehyde 3-phosphate, and then you do the steps that we discussed. Okay, that's the first. So in a sense, you build up high energy phospho compounds. Okay, so that the phosphate can be transferred to ADP and make ATP. Um, and you have a couple extra reactions because you cannot bypass, you need to bypass the toxic intermediates. Okay, so that's why you have the number of reactions you have, which includes, of course, fermentation here at the last step from pyruvate to lactate, because you have to regenerate the NAD plus and keep doing the entire lower part of glycolysis, get your ATPs and get all your beautiful 3 carbon intermediates from here on, which all can be used, or most of them anyway. These two, 3-phosphoglycerate, phosphoenol pyruvate, they can be used and pyruvate for amino acid biosynthesis, for example. Not they can be, they have to be used. To make amino acids, you have to use these intermediates. All right, so we're going to close with just a couple comments about flux, which is, the material, the flow of material per unit time through a pathway, and how much ATP and what investments you have to make for that. So it's almost like, you step on a gas to your car, and whether you want to go faster, of course, you're not going to spend a lot of gas. So these are kind of the same considerations that the cell has to think about when it decides how to regulate, let's say, glycolysis or any other pathway for that matter. So the vast majority, not the vast majority, the most common glycolytic pathway, the upper part of glycolysis, is what we described, but many bacteria, so not in animals it's the most common pathway, but in many bacteria there is a second pathway. In fact, both of them coexist. I mean, E. coli can do either the EMP pathway and then Meyerhoff-Parnas pathway, which is the standard, so to speak, the textbook version of the upper pathway, which is how we described it. But there is the Eden-Dudorov pathway, okay, that the E. coli can do both. So this is what happens. So this is only the upper part. We're not, as we discussed, the lower part is fixed. It's the same. So what happens in the ED pathway is that you have different reactions. So you get the glucose 6-phosphate. So you have hexokinase, and then you don't make it into fructose. Instead, you phosphorylate and you get 6-phosphogluconate. That's a reaction that is happening in normally in cells. We'll see it in the pentose phosphate pathway. And then, so that's a 6-carbon unit, and you split that in one glyceraldehyde 3-phosphate, which enters the canonical lower part of the pathway, and the other 3-carbon unit is straight up pyruvate. Okay, so that's the point now. So what's the net ATP production here? It turns out you use one ATP here in the upper part of glycolysis through the ED pathway to phosphorylate it to make it glucose 6-phosphate. So that's hexokinase. But you don't use a second one in PFK that we've seen with the standard EMP pathway. Okay, so there is, for every glucose, there is one expenditure of ATP. And what you get out then, you get the 2 ATPs from one glyceraldehyde 3-phosphate. So you get out 2 minus the 1 you put in. The net ATP production here is 1 ATP, right? So again, because you spend one less in the upper part. Why don't they phosphorylate to keep you know um, the products in the cell? Well, gluconate 6-phosphate is negatively charged. Glyceraldehyde 3-phosphate is negatively charged. Pyruvate is, it turns out it's hard to get out. Okay, so it will not get out. It has a carboxyl group, so it's negatively charged. So you don't need to worry about pyruvate. Now, um, that's why they don't need to spend an extra ATP. So the net ATP production in the AD pathway is 1 ATP per 1 glucose. So you do get ATP. You do everything else the same way. You get lactate, so you can do fermentation, so you can keep doing the lower part of glycolysis, but you get 1 ATP. So the question is, why bother doing the AD pathway? And as I said, there are organisms that do both. I mean, E. coli does both. When you can, why get one when you can get two? Okay, why stay with a burger when you can have a steak here, right? Well, this is the idea. Okay, so the delta G's are actually the overall the cumulative delta G for the standard canonical pathway is pretty good, about 100 kilojoules per mole, and it's even better actually for the AD pathway. Okay, it's a steeper change. In fact, it's minus 165, so it's more minus than they're both minus. They're both thermodynamically favorable, but this one is even steeper. Okay, so that's super favorable, no question, more even than the EMP pathway, chemically speaking. And but then why the 1 ATP? Well, here's why. So as we have discussed in normal, under normal conditions that let's say yeast that does the EMP pathway doesn't do the AD pathway. Okay, you have to maintain this huge flux. You have to have a huge fraction of your protein content being devoted to just the glycolytic enzymes. So that's a huge expenditure. So it's not useful to only see how much you get out of glycolysis, but what do you actually put in to make all these enzymes to get all these things happening? So this is where the ED, if you compare the EMP reactions and the ED reactions for the upper part of glycolysis, the protein cost is much higher for the, to make all these enzymes is much higher for the EMP pathway as opposed to the ED pathway. So yes, you get, 1 ATP when you can get 2, but imagine how many ATPs you spend to maintain all these enzymes at super high levels. Okay, so it's actually pretty good. And if you don't have to go super fast to grow fast and divide as fast as possible, the AD pathway is a very nice economical approach that gets you there, and in the end you're happy. So that's the idea. You have to balance the ATP production here with the actual cost of maintaining the infrastructure of glycolysis. All right, so at this point, we'll stop with this video. That concludes glycolysis and its rationale. I hope you enjoyed it. And this material also, up to this point, will be covered into exam 1. And I'll see you next time.